Prior to mitosis, thin strands of DNA in the cell nucleus thicken into chromosomes, which then duplicate themselves. The first phase in mitosis is prophase. This is when the centrioles divide and move apart. The nuclear membrane also begins to disintegrate. Next phase is metaphase. This is where the duplicated chromosomes and their centromeres become aligned in the center of the cell. Also, they are attached to the spindle fibers. In anaphase, the centromeres split, and each of the chromosomes separate into two identical groups that move to the opposite poles of the cell. Lastly is telophase. This is where the nuclear membrane is formed around each new group of chromosomes. Cytokinesis is also preformed, and this is when the cytoplasm divides into two daughter cells. Meiosis. In prophase 1, the chromosomes condense and the nuclear envelope begins to break down. In metaphase 1, the pairs of homologous chromosomes move to the center of the cell. Anaphase 1 is when the homologous chromosomes move to the opposite poles of the cell. Telophase 1 and cytokinesis. This is where the chromosomes gather at the poles of the cell and the cytoplasm divides. In prophase 2, the homologous chromatids do not duplicate, but they separate, and a new spindle fiber forms around the chromosomes. In metaphase 2, the chromatids line up in the middle of the cell, and the centrioles and asters are at the poles. Anaphase 2 is when the centromeres divide and the chromatids move to the opposite poles of the cells. Telophase 2 and cytokinesis. This is where the nuclear envelope forms around each set of chromosomes and the cytoplasm divides into 4 cells, each with half the number of chromosomes of each parent cell.